Hello, and thank you for joining us. Today we're here to talk about the autocorrelation function, or the ACF. The ACF is the key data that is generated from a dynamic light scattering, or DLS experiment. DLS experiments can give you a lot of really important information about your candidates and formulations, and that information is derived from the ACF. However, the relationship between the ACF and the actual parameters that you get from DLS can sometimes be vague. So sometimes it might feel like you're just tossing your samples into a black box and just trusting the information that the instrument gives you makes sense. And today I want to talk about where this autocorrelation function comes from and how to take a closer look at the raw data generated from DLS in order to help you make clear, rational decisions about your DLS data. I'm Dr. Leah Pandisha, an application specialist for nanotemperature technologies, and my experience with light scattering techniques over the years have shown me what information researchers need to use to make the most of their DLS data. We're going to start today by reviewing what DLS is, and then talk about the autocorrelation function and how it's built from this light scattering data. After that, we will review some fit models and how those give you information about DLS parameters of your proteins and biological molecules. And finally, we will wrap up with discussing some pitfalls you may encounter with the autocorrelation function and your DLS data and how to fix them. So please feel free to ask any questions you may have during today's talk in our Q&A chat box. It's the purple icon at the bottom of your screen. And we'll have some time at the end of today's webinar to answer any or as many questions as we can get to. So with that, let me share my screen and we'll begin our talk. Okay. So researchers express and purify proteins and viral vectors for a wide range of applications, from structural biologists studying mechanisms to biological therapeutic candidates to the development of gene therapy vectors. And knowing the size and structural behavior of these molecules and how they change in response to small molecule additives, buffer optimization, or mutational changes is really crucial to making sure that you get the best and optimal candidates during your developmental workflow. So researchers need stable biological molecules to work with during this assay development and scale up. But what do we mean by stability? So stability is a multi-parameter matrix that can cover many aspects of the biophysical characterization. But for today's discussion, we're going to be focused on colloidal stability, which is the ability of molecules to remain folded and functional while resisting aggregation. And one of the many techniques used to assess colloidal stability is dynamic light scattering, or DLS. So what does DLS tell me? DLS experiments are probing light scattering, and they provide information related to the size of your particles. So you get information on the hydrodynamic radius, as well as information on the polydispersity index, or the PDI. And the polydispersity index tells you how well your proteins are folded, or if they're starting to unfold or oligomerize. And it will also tell you if you have any aggregation or other contaminating particles in your sample solutions. So how does DLS work? All particles in solution are going to scatter light. And when we talk about particles today, I am going to mean everything from proteins to nucleic acids, buffer additives, dust and dirt. DLS measures everything in your sampling area, which is kind of the pros and the cons, right? So depending on the size of the particle, it's going to scatter light differently. Large particles that have a radius greater than 1/10 of the incident light's wavelength are going to exhibit Mie scattering, where they scatter light more intensely in the forward direction, while smaller particles that are sizes less than 1/10 of the incident light's wavelength are going to exhibit Rayleigh scatter. And Rayleigh scatter is isotropic, or it scatters light equally in all different directions. And this is important because for many biophysical characterizations, we are most interested in our smaller particles like proteins and biomolecules. So here is a quick overview of how DLS looks at scattered light to obtain particle sizing information. And I'm going to go into this more in depth later on, but for the time being, I just wanted to give you a brief overview. So when particles move in solution, they're going to move in and out of the sampling area based on their Brownian or their random motion. And this is going to cause changes in the scattered light. So DLS is probing these intensity fluctuations of the scattered light as a function of time. And those changes can then be used to establish this autocorrelation function, or ACF, which is describing the rate of these intensity fluctuations, or how likely it is that a particle is going to be found in the same location over a period of time. We then can fit this decay rate of the autocorrelation function to get a diffusion coefficient, and using the Stokes-Einstein relationship, we can then calculate information on the particle size. So interpreting this autocorrelation function is crucial to making appropriate decisions about your sample data. So let's dive into understanding where this autocorrelation function comes from and what it tells you. So the first thing that we want to understand about these particles moving through solution is that large and small molecules are going to behave differently. And this difference in their behavior is what ultimately allows us to calculate particle size, as well as look at if there are different populations of particle sizes in the sample solutions with DLS. So the smaller a particle is, the faster it's going to move and diffuse through solution compared to larger particles, and smaller particles are going to have more collisions, and they're going to change locations more frequently than larger particles. So this means that their change in location and their movement through solution is going to correlate to more intensity fluctuations in larger particles. And we can see this in the two plots shown here, where smaller particles and the intensity as a function of time is plotted here, and we see a lot of variation and a lot of fluctuations in the intensity, whereas larger particles move a bit slower, so they have less changes in the intensity. So the autocorrelation function is extracting these time-dependent intensity fluctuations and comparing the similarity of the signal intensities over time. So how do we translate these intensity fluctuations into an autocorrelation function? So here we have a cartoon diagram of some small particles in solution and its corresponding autocorrelation function, and then some larger particles in solution and its corresponding autocorrelation function. So at the initial position, these signals are very similar because nothing has happened. We only have, one time point. So regardless of the particle size, we have this autocorrelation value here shown in the yellow dot. As time progresses, for smaller particles, they're going to move more quickly through solution and have more intensity fluctuations. So this means that the signal intensity at a certain time point compared to the initial time point is going to be larger for smaller particles because we have more changes in the intensity fluctuations. Whereas for large particles, shown in the bottom, as time progresses, they're not going to move as much, so there's less intensity fluctuations, and the similarity between the intensity at a time point, say 2, compared to the initial position is more similar for these larger particles than for smaller particles. So we can see that as the smaller particles move through solution as a function of time, since they have larger differences in the signal intensities, we get a change or a decay sooner than we do for the larger particles, where they have more similar intensity fluctuations as time progresses, so that decay is a little bit longer. So now we know how we get the autocorrelation function from the intensity fluctuations of the scattered light. Let's move on to how we get information about particle sizing and dispersity from this autocorrelation function. So we know that the size of a particle is going to affect the ACF and the rate of its decay. The size affects both the speed of a particle's motion and therefore how quickly the intensity is going to fluctuate, but it also is going to affect the intensity of the light scattered at a given angle. So larger particles are going to scatter more light and have a larger signal amplitude than smaller particles, and I'll go into that a little bit more later on. So the differences in how the size affects the autocorrelation function are shown here on the left, where the larger particle is this red curve and the smaller particle is the blue curve. And we can see for the smaller particle, we have a slightly decreased amplitude compared to the larger particle and a steeper slope. So in order to acquire particle sizing information from the autocorrelation function, the raw signal is fit with a mathematical model that describes the rate of its decay to derive a diffusion coefficient signified by this d value or d variable. The diffusion coefficient, along with the temperature and the viscosity of the sample solution, is plugged into the Stokes-Einstein equation shown here. And we are allowed to, or we can then calculate the hydrodynamic radius r. Don't worry too much about this equation. It's really just here to highlight the relationship between the diffusion coefficient and the hydrodynamic radius. And then on the right, we have the intensity distribution plots, which are used to better visualize the particle size for different samples and/or different particle species. So there are two ways of analyzing the autocorrelation function to determine particle size: the cumulant radius and the size distribution. So the cumulant radius fit is for monodispersed systems or single particle species, where the radius is centered within the intensity distribution curve. This fit is assuming that there is a single population of particles, so they have a single diffusion coefficient with a Gaussian distribution about the mean. So the output value, or the radius, is the average of the particles in solution. The size distribution fit can be used for either monodispersed or polydispersed systems, since it accounts for multiple discrete populations. And this size distribution fit does not assume a certain distribution of particle sizes, but instead uses a intensity weighted distribution of multiple diffusion coefficients, which is how it's able to calculate the hydrodynamic radius for multiple different particle sizes in solution. So ultimately, the cumulant radius is taking the average particle size present in the entire sample volume, whereas the size distribution is looking at how many discrete particle sizes are present within the sample solution and at what proportions. So particle size is not the only information that you get from fitting the autocorrelation function. We also get information on the dispersity of the sample solution. And the dispersity refers to how many different size populations are present in the sample and how spread out these populations are about the sample's mean radius. So the PDI, or the polydispersity index, is the output value that we get describing this sample heterogeneity and is a useful metric for sample quality. So you heard me mention mono and polydispersed systems in the previous slide. So for a monodispersed system, shown in the example on the left, the particles in solution are a single size. They're very homogeneous. And we can see in the intensity distribution plot that there's a single peak, which is corresponding to the single particle species with its corresponding radius. And the peak is very sharp and narrow, indicating that there's a tight distribution about the mean. Whereas the polydispersed system includes a variety of different particle sizes. And in this example, we see in the intensity distribution plot, two peaks corresponding to two different particle species with two different radii. And these peaks are relatively broad, which is indicating that those particle populations are both polydispersed or heterogeneous. So here we see the intensity distribution plots of a monodispersed and a polydispersed system fit with both the cumulant and the size distribution fits, where the scattering intensity is plotted as a function of the hydrodynamic radius. So here the cumulant fit is signified by this dashed red line. It has this red line because it's assuming a Gaussian intensity distribution. So there's going to be no change in the shape of that peak because it's always assuming a Gaussian distribution. The size distribution fit, however, is shown here by these blue peak shapes. Since it's the distribution or the width of the peak is going to change depending on the dispersity of the particles in solution. So for instance, in a monodispersed system seen on the left, there is going to be a single particle species, and we see that the cumulant and the size distribution fits overlay quite nicely because there's a single species and the peak is very symmetric for these single species populations. For a polydispersed system, shown on the right-hand side of the screen, there are two different peaks in this example, which correspond to two different particle species, each with their own radii. However, the cumulant fit, that dashed line falls within those two peaks. And again, this is because the cumulant fit assumes an average radius from all the particles in solution. So in the case of a polydispersed system, you would actually want to use the size distribution model to acquire particle sizing and dispersity information. So even if there are single species present with a single radius, there still could be some variations in that measured radius if the protein potentially begins to unfold or oligomerize, or if there are potentially some different oligomeric states, but the variations in that are going to be indicated by the PDI. So again, the PDI value is giving you clues to your sample quality. So a more tightly folded protein in the figure on the left here with the same conformation or oligomeric state might have a more narrow peak with a lower PDI, indicating there's more monodispersity of those particles. The broader that peak shape is, the higher the polydispersity index is going to be, shown by the figure on the right. So that particle species is going to be a little more polydispersed or less homogeneous, which could potentially be indicating that there is some unfolding occurring or there are multiple oligomeric states. So what PDI values do you want to look for? What PDI values are considered good? So here is a list of a variety of different PDI values and their corresponding dispersities, but I really would like you to keep in mind that these are guidelines. These thresholds are kind of relative, and they're going to vary according to your application. So for instance, our threshold, or many thresholds for a monodispersed sample, is between 0.1 and 0.3 PDI value. However, your tolerable threshold might be something more like 0.2 based on the behavior of your biologic and what sort of dispersity you need to continue on with that candidate molecule in your developmental workflow. The higher the PDI values are, the more polydispersity and the better, or the more propensity there is for aggregation. So if your measured PDI indicates polydispersity, you're going to need to dig more into your raw data, like the autocorrelation fits and the intensity distribution plots, to help to decipher not only what the best fit model to use would be to get good radii and PDI values, but also to see if you have multiple species present in your sample solutions. So here we see the autocorrelation function, and the raw data is shown by the navy dots here, which is a little hard to see. And we have a cumulant fit and a size distribution fit, where the cumulant fit in teal is quite poor. However, the size distribution fit in purple overlays the raw data very nicely. And this is corroborated by the intensity distribution plots, which show that there are 4 discrete particle size populations in this particular sample as well. So we've talked about how the intensity distribution gives you information to make better decisions about your proteins now that you know a little bit more about dynamic light scattering and what the autocorrelation function is and what it tells you about your data. But I want to give a couple more real world examples of you go into an assay and at some point something's going to go potentially wrong. So how can you recognize what bad or poor data is? And not only how can you recognize it, but how could you troubleshoot it and potentially avoid it altogether? So there are 2 parts of the autocorrelation function that we want to look at to distinguish between good and bad data. This would be the amplitude, the baseline, and then of course we're going to look at the fit itself to the raw data. So in red we have the baseline, which should become linear at a specific value since it's a decay function. And then we also have the amplitude in yellow, which is the height of the signal above the baseline. And as I mentioned earlier, the more scattering intensity, the larger that amplitude is going to be. So theoretically, the maximum amplitude of the autocorrelation function is 1. However, biological solutions aren't ideal systems. So anything between 0.2 and 0.9 is acceptable, but typically you'll see something at about like 0.4, 0.5. So the first issue that you will most likely run into at some point is dust and dirt. So if there is dust and dirt on or in your sample solution, this is going to be observed in the autocorrelation function as an elevated or a lack of a baseline, as well as a poor cumulant fit shown by this red line here. And if the size distribution fit did actually fit something, then you'll have very high PDI values as well. So it's super critical for your DLS measurements because, again, they're looking at everything in that sampling volume, whether it's your actual sample or dust. So it's really critical to keep not only your samples covered and protected during sample preparation, but also the materials you are using for sample preparation should be covered and put away in drawers. So your pipettes, pipette tips, plates, vials, whatever you are prepping samples in. Here we can see an example of the intensity distribution plot of a dust-contaminated solution. So the red plot here was a buffer solution that had some dust and dirt in it because it wasn't stored properly or put away. So while it's not always critical to running a buffer blank during your DLS measurements, if you are consistently seeing some peaks or high PDI values so that you don't expect to in your regular sample solutions, then running a buffer alone to rule out any sort of contamination of the dust and dirt would be a great idea. And then you can also filter buffers prior to your DLS experiments as well. Air bubbles are going to be another thing that can cause anomalies in your ACF. So if an air bubble is present, the raw data is going to kind of like jump around. So we can see this here in the purple data points. It's very erratic, so it's going to lead to poor fits as well. The signal intensity might be very high or low depending on where in the air bubble the measurement is being read. And there might also be an increase in the scattering intensity for a single acquisition compared to other acquisitions for that particular sample. So some things to fix air bubbles is to centrifuge your samples prior to loading to remove any small air bubbles, as well as not vortexing or shaking your samples prior to your sample loading or measurements as well. If aggregation and sedimentation occur, the ACF is going to have a very high signal intensity and possibly a higher amplitude than expected at a given concentration. So large particles aren't moving, or they're moving really slow. So your decay is also going to look very gradual, rather than that sharper decay we've been seeing in the previous slides of the autocorrelation function. You're also going to see an elongated shoulder of that decay and a baseline that never plateaus. So some things to fix aggregation sedimentation is to centrifuge your samples and then transfer those samples to a fresh tube prior to running your DLS experiments. You can also filter your samples with a 0.2 micron cutoff filter. This is going to be dependent on the type of experiments you're running though. So if you're specifically trying to see if there is aggregation in your samples, then you wouldn't want to filter them. And if aggregation persists, then you would want to reconsider or consider reformulating your buffer since it's not ideal for this particular candidate molecule. And lastly, autofluorescence. So this is going to be at the same wavelength as the detector and can create poor autocorrelation functions as well. These are seen where there's really no decay during the acquisition and generally some very high scattering intensities. So while this is quite rare, if it does occur, the sample is not suitable for analysis at the DLS wavelength being used. So with that, I have some key takeaways from the talk today. So DLS measures the fluctuations of the scattered light intensity, and these time-dependent fluctuations create the autocorrelation function, or ACF. The autocorrelation function decays quicker for smaller particles than for larger particles, since smaller particles are going to diffuse through solution faster, changing their locations more frequently and therefore having more intensity fluctuations. The autocorrelation function is fit using two different models, the cumulant radius and the size distribution, to build an intensity distribution plot and derive information about the hydrodynamic radius and the polydispersity. And the model and the data used to fit your system is going, or fit your autocorrelation function, is going to depend on whether your system is monodisperse or polydisperse. So cumulant fits are used for monodisperse systems, whereas the size distribution fit can be used for either monodisperse or polydisperse systems. And the polydispersity index provides insights on sample homogeneity and quality, as well as whether the sample is potentially unfolding or oligomerizing. So I hope this talk has given you a better understanding on how DLS is used to probe colloidal stability, the kind of data you can get from these experiments, and then how to analyze them, as well as how to prevent and troubleshoot potential issues with your data, which ultimately will hopefully allow you to make better decisions and move confidently forward with your biophysical characterization and assay development.